Hello friends, we have again started a new video and this is regarding a fungus. Again this is regarding a fungus. This fungus is found in the Malabar spinach. This is a green spinach found in locally in West Bengal. This is called pui shag. So this fungus is these spinaches are very well and very nutritious to eat, nutritious vegetable. But what happens that when it is not grown, or it is not, if it is not protected during its growth in the cultivation, it catches a fungus, and this fungus is actually is called. Sir, this fungus is actually, you will see, able to see in circular patches, small circular patches on the spinach leaves, and these are Cercospora betiola, Cercospora betiola, and this is literally looks like a circular patch, circular ring, so it is a fungus, and these funguses are very bad for human health, as you can see, as you know. So therefore, these funguses are not to be, these spinaches are during that spinaches it should be cultivated very carefully. Anyway, we are going to see this fungus under laproscope. First we will see this fungus under laproscope, then this fungus we will put into the culture media. Here we are going to use an hectoin-enteric agar. This is a special kind of agar, hectoin-enteric medium, actually hectoin-enteric medium. So this is a specialized culture media. We are going to use it, and we will cultivate this fungus and to see this morphology and all that, as usual in like the previous videos, and we will do it. So let us begin the video and the process of cultivation, process of scalping the fungus, excavating the fungus. Before that, we will be visualizing in the laproscope. So after that, we will going to see the process of putting it into the culture medium. So this I will show you in details, and you will be able to see how it is done. This how it is put in, excavated, and how the culture media is put given in the culture media. So that we will see. So let us carry on. So as you can see, the Malabar spinach under laproscope, as you can see, this is Cercospora fungus. This is the brown patch, brown, whitish, pinkish brown patch. This is the Cercospora. That is the big Cercospora spot. You can see in the laproscope. You can clearly see in the under laproscope. That is the fungus. That is the fungal patch, and that is the actual fungus. So we are just trying to focus the laproscope, studying the laproscope, and you can see the fungus. So this is the fungal part on the leaf. You are just seeing the surface of the leaf, and you can see the fungal spots, other fungal spots are there. So that is the laproscope, and that is the fungus, Cercospora fungus. The whole leaf has been attacked by this fungus, so you can well see from this laparoscopic view the other parts of the leaf. We are just visualizing some Cercospora has cracked. Actually, you can see a crack of Cercospora. The leaf has cracked due to this Cercospora infection, and that is another oozing Cercospora. This is a big Cercospora, again a big Cercospora, and that's it. So friends, we are going to excavate the fungus, and as you can see, we have made an arrangement of the scalpel and the forceps. So we are going to extract this fungus. This is a very spot fungus, so very less amount of the vegetable matter tissue is needed. So just we are making a square incision, giving a square incision to the leaf, and just taking the fungus out. This is very easy, just to scrap the leaf with a scalpel, little strokes of the scalpel, and it will come out. So just the sample will come out, and big amount of tissue will come out, and little bit we will take, and that is the big tissue. So now you are going to select more spores. Just let us see, more spores are there. It is full of spores, so let us take more, two or three more spores, so that we get the fungus out. And this fungus is there. So the fungus is there. So let us see anything else we can take from this leaf. So here are the two. We can take these two with a long incision like this, and you can see the incision is done. Just taking with the help of the forceps and putting it in the culture. So this much is done. So our excavation is ready. So now we will put it into the culture media. So as usual, the same thing. We will take one pipette and steady pipette, and now we will just take on the steady pipette and just switch on the pipette and take some of the culture media, around 6 ml. The culture flask. So this is red in color. The solution is red in color. It is a chromatic indicator, almost a chromatic indicator. So just this much we will take, and it is in the culture media. So it is almost red in color, crimson red in color. So that would be, and our spinach, our culture is ready. So now we are going to work in the usual manner. We will take a folded tissue paper and take some cotton plugs. Plugs, and our culture is ready. So this we will going to inspect under TCM 400 microscope after 5 days. So that's all for now. We will be putting it into the incubator, and we will be observing the Cercospora under TCM 400, and also we will make a fluorescent stain, and we will make the Cercospora under fluorescent microscope. So regarding that, I will be explaining the fluorescent staining process, the mycotoxin extraction process, and also the culture process. So goodbye from now. We will see after 5 days. Hello friends, we are back after 3 days with the fungus of Cercospora. It has become, after 3 days we've seen that it has become have made a considerable growth to study. So to study the microscopic examination. So that is good and bad both, because good means we can study well, and bad means it is within 3 days it has made a considerable growth. So this is a very fast growing fungus. So let us see, after we are taking some 3.5 ml in the petri dish, and we will examine under the TCM 400 microscope, and we will see the morphology of the fungus. So let us begin the experiment. Just we are going to take around 3.5 ml of the fungus culture media, and we will see. So let us see. So this is our culture media. It's a profuse precipitation and profuse accumulation. So let us begin the test. And this is, you see the fungus, you can see the microscopically when the fungus is grown. This is the You can see the macroscopic growth of this fungus. So let us see how it is done. So let us see how it is done. So that is this. That is this is, you can see, the Cercospora fungus, and you can see the conidias and the conidioforms, and the base of these conidioforms, the base of these tentacles are stromas. So it is clearly visible in this fungus. You can see the tentacle-like structures, though not very clear, because this is we are viewing directly on the TCM400 microscope, and the culture media is very thick, so it is little difficult to estimate. But still, the conidias are visible, and the conidioforms are also visible. So later on, we will try to see it is to make it more visible. So let us see what is in there in the solution, the culture media, and we are moving the culture media petri dish, and we see more conidioforms are there. You can see the conidioforms. This is the main fungus of the main fungal part of the Cercospora. So you can see the tentacles, the conidias and the conidioforms, and the stroma is also visible. The base of the fungus is also visible. The central base circular fungus, central base area, the central area is called the base of the fungus, or the stroma of the fungus. So this is visible, you can see. So this part is also visible. These are the mycotoxins. The micropores are released. So this consists of mycotoxins. So this is also another part. So these are the stromas. So you can see. So here is the main fungus, main system fungus, the main fungus, and you can see the cercosporas. Here it is very clearly visible. Here is actually it is visible. You can see the cercosporas. These are the cercospora. These are the conidias and the conidioforms, and the base stromas are visible. So this is the main fungus, main body of the fungus. You can see it is a 100x magnification. It is clearly visible. So you can see what is now clearly visible. So this is almost, we are end of the slide, and this is also the another part of this fungus. Now it has become very, very sporadic. This fungus has become sporadic, and it has profused. So friends, this is our Cercospora under fluorescence. You can see the circular spots, and that is why it is known as Cercospora. This is under fluorescence 10, and you are utilizing under 400x magnification. And so this is the orientation of the fungus, the pattern of the fungus, the fluorescence pattern of the fungus. They are found in groups actually. You can see the Cercospora groups here and there. So this is actually the circular spores. So this is the structure of the Cercospora fungus. So this is what you can see in the slide. This is under 400x magnification, and this is the other portions of the slide. This is going on, and this is the Cercospora fungus under fluorescence microscope. It should consist a mycotoxin of cercosporin, but maybe more mycotoxins are present. It should consist of cercosporin, but maybe more mycotoxins are also present. So that will be revealed under UV spectroscopy. So that we will do on the next part of the video. Till now, this is the Cercospora in front of you in fluorescence staining. So the visualization of the slide is almost over. So this is our Cercospora. You can see the geometry of the Cercospora fungus, and this is all about Cercospora baeliota under FLR. So you can see the UV spectrum of the Cercospora fungus, and as usual it is consisting of cercosporin, but 15 more mycotoxins are present. Total 16 mycotoxins are present in this fungus, So therefore usiloxin is present. All you can see in the left hand side, extreme left hand side, the usiloxin curve is there, and the rising, all the rising curves, all the absorbance peaks are there. The small organ absorption peaks are there. So total 16 mycotoxins are present, including cercosporin. So this is the condition of this fungus. It does not only contain cercosporin, it also consists of more funguses, more mycotoxins. Usiloxin is present and all other trim organs are present. So this is the condition of the fungus, as you can see, and it is a very bad type of fungus, as the spectrum reveals. So this is all about Cercospora fungus. You can see from the UV spectrum.